universities in Ghana, in Ethiopia. And these were not just sort of handshake deals. So there had to be money put down and investments made. This particular uh, relationship was led by Kevin Chen, who's a hand surgeon, very famous hand surgeon at the University of Michigan. So I got to go because critical care was supposed to be one of the things that was going to be explored. And so I went and I met KFC, who was in the emergency department, and uh, just super crowded ER, and toured there and their ICUs, and it was just so similar, the problems we were trying to sort of co-solve. So, detecting sepsis and epidemiologic, outcomes, ventilator associated pneumonia, ARDS, just a lot of different things. But we never really were We never were really able to get something jump started. And then out of the blue this year, KFC contacted me and was able to arrange a 2 year. I don't know, what are you calling it? Not a sabbatical, huh?
I
A
don't know.
2 year fellowship,
Yeah.
let's say collaborative training through Global Reach, through the University of Michigan, and his concentration is going to be data science. So, in addition to his medical degree and residency training in emergency medicine in Taiwan, uh, KFC recently received a PhD from Johns Hopkins in clinical investigation and did a sort of like a data science fellowship there. And so we were fortunate to get him sort of directed towards us. So it's going to be 2 years. He started officially, I guess, in August or July. Just got settled in, brought his family here. And so we want to sort of introduce him to the ecosystem through giving talks, having him spend some time in various people's labs and shops, learning what they're doing. But it's uncanny when he showed me the list of things he was doing, which Jay is going to share, just how much overlap there is. Everything from signal processing and sensors to imaging to physiologic phenotypes and things like that. And so the problem may be trying to narrow this down. So we want to invite him to give a talk to the greater Weil sort of data science ecosystem. But we thought it would be good for him to sort of introduce his work, let us take a look and see what the membership would be maybe most interested in, because we do want him to really develop some robust collaborations. We've added him to our IRBs. He's meeting with the data science team to sort of understand what's possible. But again, you'll see everything from sensing and signal processing to inferential causality. So I'm going to stop right there and let KFC sort of take the wheel, introduce anything that I've neglected to put on. My introduction to the bigger group when you speak will be more polished than this. And then Jay, I don't know, Jay, if you want to share the list of things.
Yeah, and I think everybody could see it. I put it in the chat. It's also in the meeting invite. It's a high-level list that we kind of broke down by research topic. If there's anything of particular interest to you, please feel free to put it in the chat so that Dr. Chen can see it. I'll make note of it, and we'll spend a little bit of extra time when he goes over it today, because he has a very large collection of slides. Not all of which we might be able to get through today. So we want to kind of focus on the topics that are of most interest to our group here, and then use that kind of reception that we get to tighten up and hone in on a real interesting presentation for the broader community in October. So go ahead. Thank you.
So I guess while you're still looking at the PDF, the spreadsheet, I'm going to start my high-level introduction of what we've been thinking about and what I think is worthwhile for us to collaborate. So I think the title is just to attract people that we are trying not only to do some research, but also trying to deploy those models in our real world setting. So I think Kevin have introduced me a lot, but previous I was trained at Hopkins. Given that period of time is biostat and epidemiology training, so focus more on causal inference, focus more on data analysis. But at that time, I don't have the fortune to get exposed to deep learning type of approach. But to be honest, I'm as trained as a clinician. Math is something that is really far away from me. So sitting in Professor Najarian's class is really a luxury for me to really sit down to review something. But now I'm focused more on the utilizing this approach on both the biodynamic type of analysis to reinforcement learning type of thing. So I'm going to deeper to introduce you. So this is the web page of my lab. We focus on prediction and also the outcome research. So that means we are not stopped by merely prediction, but also want to see if the prediction can have a clinical impact. So this is the introduction of our pipeline. We start from data collection to storage, analysis, and then the deployment. So we utilize not only structured data, but also unstructured data. And in the past, when there's no deep learning, we use regular extraction to extract some text information. And then now we also have other tools available. So let me just introduce our hospital a little bit more. The hospital actually is a system which has, I think, is more than 8 branches island-wide, because we also have some hospital run by us, but is owned by the government. So interesting, we have a unified homebrew EMR system, and we also have very large ED volume. As you can see, after the pandemic outbreak, the volume just getting bigger. In Lincoln branch, which is the largest one in the north, it actually has more than 16,000 visits per year. So we also have a unified research database. In this publication, this paper, one of our researchers managed to compare our database with the, I think it's a relatively well-known national claims database, which is our National Health Insurance Research Database. So we have of course, we cannot be the coverage is not like 100%, but our clinical information or the standardized data format is at least as good as the national claims database. And I also have experience analyzing this database in the past. So take the COVID severity index, for example, because at that time we think this is one of the very simple score that we could apply for our colleagues in the clinical setting. So we managed to use 1 month from scratch to application. Although this is a relatively simple score, we still need to extract the information such as the O2 flow rates from the unstructured data. And then you can see here, although it's probably all Chinese, but the red one just indicates the severe COVID-19 infected patient. So we deploy that to our EMR to help clinicians to stratify their patient. And furthermore, our institute is trying to adapt the HL7 or FHIR compatible structure nowadays in order to achieve the goal to get into the smart, sort of like the EMR version of the App Store in the future. All right, so that's the introduction of our institute, and I'm going to use sepsis as one of the scenario that I think we could just use this multimodality and reinforcement learning approach to help our real-time decision making. So I'm gonna spend most of time on the non-invasive monitoring, posture detection, and the phenotyping type of approach, and then I hope I can still have some time to describe our effort in deep reinforcement learning and contrast learning. So start with the non-invasive monitoring. This is just a screenshot from the Edward ClearSight device that we utilized in the past. And then as a clinician, we know that there are some available tools to predict response to fluid. As you can see here, the passive leg rest test has relatively good positive and negative likelihood ratio to detect the need for fluids for patients with hypovolemia. And then in an updated review article, some researcher also managed to find some subgroup that this technique specific maneuver will be helpful to identify the need for fluid resuscitation. But this technique is still really cumbersome. It's not easy to do in the ER. To be honest, it's not easy to obtain a cardio outputs, and then you need to briefly raise the leg and then get the change of the cardio output type approach. So we've been thinking maybe we should just utilize the technology, use the post-contour analysis, for example. I'm pretty sure many of you must be familiar with the technique. And with some formula, we can calculate some other parameter of the hemodynamics. And then this is just one example that we can collect patient data for many minutes or hours. But we also have many issues to tackle, like the missing data or some outlier issues. So that's why I want to really have the opportunity to work with you guys. And then using this data, although we are not happy with the quality of the data, we managed to develop some model to predict septic shock using this data. And this is a manuscript in preparation. While we are still preparing this, the data, I think this is a side project, but this is part of my plan to train our team member to be familiar with the time series analysis. So this is one example that two of my students, one is actually medical students, and then they managed to find different novel model, and one of the latest is the one released by Amazon. It's actually take advantage of the large language model and then use the time series approach. And then you can see in our prelim data, the one-shot forecasting is doing much better than the other model, which we spend like hours to fine-tune their hyperparameters. So in this specific project, I think there are some research questions that we might be able to work together. One is the frequency interval of what's the optimized number for this monitoring method. And the second is our patient is always under many different medication. So maybe we want to do some group analysis or try to see if the model is doing well, like the review article illustrate that it's not like one size fits all. We need to figure out how to fine-tune the model in different population. And lastly, I think we are not satisfied with merely predicting shock, but also predict the response to therapy. All right, so moving on to from the non-invasive monitor to specifically focus on HRV type approach. I know many of us share the same interest. So this is one of the method used in the hybrid methods. It's actually published in 2020, and they use the architecture, the CNN architecture, along with the R interval feature, and they have pretty decent performance in terms of identify the ECG nature of the ECG. So as an illustration, we have the capacity to clean the data, and then find out the appropriate package to do the segmentation, and then classify each heartbeats, and lastly, find out some important HRV features that we obtained from the previous manuscript. So this manuscript recently got accepted by the Biomedical Signal and Processing and Control. So that this is one of the examples that we have the experience, but not much, I have to be honest, to work with this kind of signal. We also use this type of approach to see if we could apply on our data set. This is a relatively smaller data set. We managed to use the algorithm to fine-tune a little bit and then to identify a patient with influenza, but it's not doing really good for COVID-19 infection. So moving on, we are not satisfied with only developing a model, but try to test if the algorithm can really help our patient. So this is one of the intramural funded clinical trial. It's already been funded, so the current status is in the Taiwan FDA. So because we need to get approved by the Taiwan FDA to initiate this clinical trial. And one of the biggest challenge, or the thing we want to try differently in this trial, is to obtain informed consent from clinician, or I should say attending physician, instead of our patient, because we believe that the AI HRV algorithm should be utilized as a less than no more than minimal harm intervention, and then only the clinician will be, influenced by this type of approach. So that way we could just manage to get a bigger sample size. So the specific research question in this project will be, we could just sit together to try different algorithms, do some experiment to see what's the optimal pre-processing algorithm for either very simple sinus ECG or some other type of reason. And then in terms of signal processing, I am hoping to explore more possibility, not only in the conventional domain, but also some novel domain proposed by other researchers, such as energetic, informational, geographic, and invariant domain. In addition, I think we can just try to convert the signal into tempogram or spectrogram, and maybe we could just apply some other deep learning method on these figures. All right. So moving on to the phenotyping type of project. This is again another very crude initial pipeline we have. After pre-process the data, doing some imputation and standardization, our methods currently is try to focus on autoencoder and then utilize a latent layer to do some unsupervised learning. And then we have a list to try on different type of unsupervised learning, and then try to find out a specific phenotype that could be utilized for cognition. So in the past, I have some experience using PCA type approach to tackle with the emerging pathogen in the past. That's in the era when sequencing is still expensive. So we applied the DNA extraction and PCR on the pathogens, followed by the electrospray, mass spectrometry, and then we analyzed the signal and managed to use the base pair as the single unit, the base units, and then once the pathogen or a specimen is out of the cluster, we could just try to explore. And this platform is actually one of the very first one, and actually identified the second 2009 novel H1N1 influenza virus infection. In addition, we also have experience applying K-mean in longitudinal follow asthma pediatric cohort. In this specific project, we managed to identify 3 different clusters and then explore their allergy profile. So In another database I've been using very extensively in the past is the collaboration between my previous mentors at Hopkins. So using, again, the autoencoder type approach along, coupled with supervised machine learning, we managed to improve the clustering, as you can see in the figure, that before and after the autoencoder embedding, the cluster become very significant. So some visualization to explore the cluster, and then lastly, what interests us most is, interestingly, the machine can identify the cluster with different severity, although we didn't tell the machine to do that. As you can see, the evidence of pneumonia or the severe composite outcome is very different in different clusters. One of our collaborators in the National Yangming Jiaotong University also are interested in doing clustering analysis among patients with sepsis. So in this specific project, they started with a mimic data set, and then they obtained data from the first 6 hours, including demographic lab tests and comorbidity vital signs, and then try to predict the patient outcome, which is the in-hospital mortality. So we use not only single point data, but also time series data. But in this specific project, we just use time domain to summarize the variation of these data sets. But we try to use the autoencoder type approach to find the best cluster in this data set. This is a very busy figure, but you can see 4 different clusters can be associated with different organ dysfunction. And then we found that the both the dynamic and static feature with the embedded approach will have the best clustering. This is a survival curve. As you see, the right lower figure just illustrate what I just mentioned. This approach really help to cluster our patients. And interestingly, if you still remember the alpha phenotype that we identified in the previous slides, and a patient with the highest SOFA score will actually do better in this clustering. We are still in the process to identify the reason in terms of what does treatment they receive. And our hypothesis is could be that the clinicians are notified by the patient status, so maybe more intervention are utilized among these patients. So again, this is one of our collaborators. We've been working for almost 10 years. So she has visited MIT in the past, so she still has some connection between the faculty, both in MIT and the Harvard University. So she's not only interested in developing model, but also interested in implement model in the EMR system. Okay, so the specific question I think we can ask in this project is the feasibility to recognize normal phenotype, and then how to translate those phenotypes in causal inference. And lastly, maybe the possibility to design trials according to the phenotypes. All right. So the next one is the phenotyping, but now we shift gear from the structured data to unstructured data. So interestingly, the medical records written by clinicians in English, although the English might be different from clinician to clinician, but we still have the luxury to extract some feature among these texts. So one of my postdoc actually developed a regular expression approach to identify those symptoms among the texts. And another interesting thing is, although the physicians are using English, our nurse are using Chinese to record their nursing records. So this is one of the group in the Academia Sinica, which I'm going to introduce you a little bit about. My another collaborator there is to use a very similar approach but different language on the nursing records. Another thing I think is important for symptoms is the dynamic nature of the symptoms, because we know that the symptoms change all the time from species to species, also from subclade to subclade. So for this specific project, a manuscript, actually we still utilize the data set obtained from the collaboration with faculty at Hopkins. We managed to dig deeper to see maybe the time is important issue that we need to pay attention when using symptom as a biomarker to predict infection or severity. And then in this specific manuscript, we managed to illustrate that day of illness is also an important feature to be used as a feature to predict influenza infection. Although we are not specifically satisfied with the performance, but as I mentioned, that maybe we just need like a cleaner or bigger database to let us do more subgroup modeling. So in addition, we also use this idea for our patient with suspected sepsis in a prospectively collected cohort study in our hospital. In this study, we are not only using symptoms, but also using some biomarkers, so the inflammation profile could be evaluated in this type of patients. All right, so moving on from regular expression to now large language model, we know that there are several medical associated language model available, and their parameters or tokens can range very widely from, million to billion. So we tried to use the Metatron early this year using their 60 billion version to see if we can use the symptom to identify patient with influenza infection. But we managed to demonstrate that GPT 3.5 at that time is actually doing better than Metatron. So perhaps the larger is better. But on the other hand, we haven't really cleaned up the data set at that point. So I think that's still too early to conclude the finding. So here I'm trying to introduce another colleague who's actually from the Kaohsiung, from the South Chang'an Memorial Hospital. He's actually also here. He's working with Dr. Vidish Warren. So they are trying to use the nodes to identify kids with Kawasaki disease, a disease that he's been studying for like years. Another collaborator is Dr. Ku. Dr. Ku is a research fellow in Academia Sinica. She's been focusing on NLP research since 1998. So I also sit in her class in our Chang'an University early this year. We've been discussing the possibility to work together, and we're currently working together for a specific project, which I'm going to mention a little bit later. All right. So that move on to applying language model not only on clinical notes, but also on the other field like the biology type of project. So during the pandemic outbreak, we also try to utilize the image transformation to see if we could just do some clustering to identify some novel emerging infection. So this is specific effort is trying to convert all these sequences, encode them, and transform into image. And after doing that, use Disney model as a filter, and as you can see here, we can just successfully identify different variant of concern. So the variant of concern could be identified as well as some lineage. utilizing further machine learning approach. And then moving on is the large language model. Now we have the genome scale language model, or in short is GenSLAM. So GenSLAM is one of the first whole genome scale foundation model. It's been pre-trained for millions of gene sequences, and then it's been fine-tuned for SARS-CoV-2 specific model. It has like lots of parameters, and the goal at that time is only the variant of concern, and it's a hierarchical transform-based model, etc. So the goal or the future direction is to use this type of approach to generate new sequence, and hopefully could be used for some preparedness in the future. So briefly, it just use some transformer plus attention type of architecture, and then use semantic bandit to do some apply some GenSLAM approach for this signal. And this is the one they have for the cluster analysis. And working with one of the engineer in NVIDIA, who's also pursuing a PhD degree in the National Taiwan University, we proved that we could just use some of the computing power to detect some of the specific strains. And this is doing specific good because the strains are so different. And then moving on to very closer or similar coronavirus strains, is the model is doing relatively good. And then using supervised type approach, we can also identify different pathogens, because we know in the future we won't have label for the emerging pathogen, very similar to my previous mass spectrometry approach. So the collaborator here will be Professor Yunong Gong, who's also a very good friend of mine. We've been working together for phylogenetic type of analysis for more than 10 years. And he's associated with the research center. Actually, I'm also associated with the research center for emerging viral infection. So the specific interesting project he's proposing is to use the big data nature of the metagenomic data, and then along with the pipeline, and hopefully can have some novel virus detection in the future. So the specific question we're trying to ask here is first the possibility to obtain some useful features for syndromic among the text. And the second one is the feasibility to generate sequence and then predict novel or emerging strain in the future. All right. So a little bit about our activity approach with collaboration between the Quanta company, which is an OEM company, previously produced lots of notebook for a company like HP or I don't exactly know the other company. But they do have a MOU with our institute that they are not interested in patent. What they are interested in is the technology transfer. So they will be the one that willing to apply our algorithm into their hardware. And then currently they have this device, this MM wave radar, specifically to detect patient activity like falling, or to use as a sensor to detect the vitals. So we take advantage of their device and try to obtain some data sets, and then have very initial premature analysis experience, which is we think we still need some help, is to do some dynamic detection. And then using dynamic radar plus color Doppler, we now can better identify the patient and their activity, although it's still premature. So the specific questions for this relatively smaller, but I think has more potential, is to first to see if we could accurately use the radar to obtain some vitals. And then, is the vital good enough to be used for variability analysis? And of course, lastly, to use that to predict patient-centered outcomes. Okay. So moving on to one of my recent efforts, which is another, I think it's another not too easy projects to apply reinforcement learning type approach to real-time, provide the real-time decision making for clinician in terms of the fluid and inotropic dosage. So we collaborate with one of the our previous colleague in Chang'an, now are having a new high appointment in University of Virginia since last year, I think. And then we also one of my master students working on this project is to see if we got not only apply this type of approach, but also obtain using the data obtained from our data set. So this is one of the algorithms that developed from researcher in China, and they used deep reinforcement learning along with the human expert, try to come up with a real-time decision for the fluid and inotropic dosage. So they use some method for feature selection, and they determine the trajectory of these variables. And lastly, they use a different network to determine the best policy along with the human expert decision. So we don't have the computing power, but we reduce the sample size a little bit to 10,000 sampling. And then we have very similar distribution, initial distribution of the inotropic and fluid distribution, but we fail to have a similar outcome. But to be honest, the model produced a different result from time to time. So the initial conclusion could be the model is now, could be the model issue, the model is not very stable, could be the data set issue, because we also figure out that many of the pre-processing algorithm provided from different researchers for the very famous MIMI data set is not really clean, because some of them even contain some missing data imputation, which is very obviously incorrect from the clinician's perspective. All right, in addition to the Chang'an Research Database, I also have experience analyzing the National Health Insurance Research Database, as I mentioned. So in this study, we longitudinally follow the patient with severe sepsis and then identify several potentially useful intervention. So in the past, blood transfusion is encouraged, but we identified that the specific blood product would be helpful instead of the whole blood transfusion. And then we also identified that
if you,
yeah, sure.
Dr. Chen, do you mind if I ask a question? I just want to make sure that we also get to discuss some of the projects since we only have like 15 minutes left. Okay. The last project that you discussed in reinforcement learning, and you mentioned that you combine reinforcement learning with human. How did you do that?
Can you just rephrase your question? Combine with what?
So I think here you said that you combined the recommendation from the reinforcement learning agent with the clinician.
Okay.
So how was that done? Was it just like, the prediction go to the clinician and they just either took it or rejected it, or how did it work?
That's a very interesting question, because in this specific paper, in specific study, researchers in China, they actually just merely adopt clinician's decision for a certain SOFA score. Like patient with a lower SOFA score, they will just adopt the clinician's practice. It's sort of like a hybrid type approach, but not really incorporate the clinical, like the human expert into the model. Did I explain myself clearly? Because my imagination is like for clinician, the difficult part is not like patient with severe sepsis, but the patient with seem to be mild sepsis. That's my argument. We should instead focusing on those difficult patients, because these therapeutic intervention is not totally harmless. More fluid could be resulted in more edematous of the tissue. But to be honest, their approach might not be the best approach, and we are still doing some research to obtain other type of approach in terms of how to, sometimes the best way is to train or to model our reward function, because their reward function is also very simple. It's using the SOFA score that generated by a specific group of patients after specific treatment combination. Did I answer your question?
Yes. So did I understand that when the SOFA score was low, they used the clinician, and then when the SOFA score was high, they used the model, or
yes,
is it the other way around?
That's the way they did it. But I don't 100% agree with their approach, and we are also trying to do the opposite way to see if the patient outcome in their study they used mortality as the surrogate of their performance of their model, which is really decent because they successfully reduced 10% of their mortality. But to be honest, we failed to reproduce the results.
So this is the clinical trial?
No, this is just an observational study. They only used the MIMI data set. Yeah.
Okay.
I'm going to speed down a little bit to save some time for discussion. So again, the database we've been used, we can do some geographic information type of approach to identify some discrepant in our country. So here is one of our collaborator is in the National Health Research Institute, and she was previously faculty in the China Medical University Hospital actually in Taiwan. So they are one of the biggest hospitals in the middle of Taiwan. So we managed to validate our model on each other's hospital. So I think she's also one of the candidates to collaborate. All right, moving back to Seer network. In this slide, I just want to illustrate that we, in the past, not only doing the machine learning, but also try to do some causal inference. And this is specific study is related to patient with influenza B infection. We identified that co-infection could be one of the important risk factor for severe outcomes, such as hospitalization and pneumonia. So, moving forward for H1N1 influenza A virus, in this specific study, we found that 6B2 could be the reason that patients are having severe outcomes, such as pneumonia. And then in this study, we successfully used the recursive partitioning algorithm to generate propensity score and use the IPTW to tease out the possible confounding. Then the last one is our recent publication in Lancet Microbe, is to evaluate the reassortment among H3N2 viruses and the severity in 2017-18 season. So our collaborators in the lab, they find reassorted virus among the patient, and then they figure out the virulence of these pathogens. And then on our end, we apply very similar the machine learning-based propensity score, and then we use the overlap method to tease out the possible confounding, and we successfully identify the possible association between reassortment and the severity. So, specific question in this project is to see if we can just make a stabler reinforcement learning, or like Sadari mentioned, the reward function is also something we want to really try to manipulate. On the other hand, we want to use not only the reinforcement, but also try to see if the causal inference can be made in this kind of analysis. All right, so the last one is the fundoscopy project. In this project, we managed to obtain some longitudinal followed fundoscopy image, and then we try to see the association between patient with cerebrovascular disease. So again, we illustrate that we are capable to process the data from the image to regional interest, and then quality control, use different filter to do pre-processing, and lastly extract the vascular structure for prediction. So it's still very early. So the initial stage is to see if this approach could be utilized to predict gender. Apologize for the Chinese prelim results. We still have some trouble, having a stable result, and perhaps it's because the data set is still small. All right, so in this project, we collaborate with the ophthalmologists who also have some experience in AI modeling, as well as a neurologist who's actually a researcher I work very closely. And this is one of the projects we have in the past. I'm going to mention that briefly just in a second. So this just very prelim, and I'm hoping to find interesting collaborator to see if you can use foundation model to do other type of approach, and then if the longitudinal follow the image can be useful. All right, I guess that's the last one. I'm going to spend just 3 minutes on this, but this could be a big one. So the less feasible, I think, but more promising perhaps, because I think nowadays the institutes are very protected in terms of data. So the data set is not easily released to other institutes. So in order to collaborate, some researchers advocate to develop digital twins, and then which could just disseminate the data set without the compromise to patient privacy. So one of the emerging companies is releasing this information, saying this kind of concept can be used for many different goals, and then it could be applied for different applications such as patient monitoring, or epidemic management, or hospital operation, etc. And then, of course, there are some challenges. The data privacy could be one issue, and accuracy also could be another issue. And while we are implementing this type of idea, in the operability and scaling would be another issues. And some institute already planning, Cleveland Clinic is one of them, to use digital twins to do their daily hospital management. And then in this scoping review, they mentioned that the rising literature, and then different resource to collect data, and different way to apply data. So very similarly, we apply the contrast learning approach, try to do some contrast learning, self-learning, because we have so many data out there, but the labeling is so expensive. So we are trying to see if this type of approach, very similar to the CLIP type approach, could be helpful. So we identified two algorithms. One is the convert, the contrastive visual representation learning. So every image has the reports in clinical setting that's 100% sure. And so then we managed to prove that we do have the capacity to implement this type of approach. But some researchers also mentioned that since the medical image is very different from the daily image, so like lots of things happen in a very small region. So some of the researchers they use the fine-grained vision language, and they try to map the region attribute pair first, and then learn the representation. So we are also in the process to do this type of self-learning, try to see if we can just label the data more efficiently. Then the following, just like illustration, we can do the image preprocessing for MRI image. So the next step is to extend the chest radiograph to MRI image in our data sets. So we do have some data to be able to use now. And then we also have some experience for ultrasound image. I'm going to skip this, but just illustrate that we can also apply these. So one of the researchers is again a trauma surgeon who has experience in image and also interested in signal processing. Another one is also newly appointed in our College of Intelligent Computing, who's actually very savvy in terms of the data preprocessing and lots of modeling experience. And last but not least, is one of the experts in the deepfake learning in actually in the south in the Chenggong University. He wants a lot of awards, and then he has various experience doing some medical signal analysis, including image and the signal. So we are closely collaborating to see if we are able to develop something for the maybe not only digital twin but also data twin. All right, let's just conclude my my report. But the specific question in this is for first with extension to other modality, and then self-learning, and then digital twins. I'm gonna stop here to save some to see if you can give me some other feedback for next month or maybe for further discussion.
So one general question. First of all, great job, Kuan-Fu. I see a lot of possibilities for collaboration. We can discuss them in more detail. But one sort of major question I have is that some of these studies, they created based on the data that you have available, and collaboration to the next stage may require some sharing of the data. What are the possibilities of sharing data for some of these projects to actually create collaboration?
All right, that's a very good question.
And if I may build on that, in addition to data, what are the possibilities of running models, for example, in Taiwan at your hospital?
Okay, so these are like twins questions. So the data itself, because not all data are restricted, because we do have consent from our patient for some project. And the other thing is we also, of course, we need to extend the IRB to be able to send the data out here, because I'm physically here in Michigan. So for some data we already have them, and we can still analyze data either here or remotely by my team, because I still have like 2 postdocs and 4 data scientists. They still work in our hospital, in our university. So we can just do modeling in parallel. And on the other hand, Tadar just mentioned that it's possible, just like the pattern I collaborate with the researcher who previously worked in the China Medical University. So the China Medical University also is not releasing their data to us. So the way we can do is to use the federal learning type approach to fine-tune our data in their institute. That's the plan. So I guess for some data, for most updated data, what we need to do is to go to the center, and before we really have the digital twin type approach, we still need to physically go to the center in Taiwan. And I do have several data scientists who has experience working inside the center. Oh, I hope I answer your question briefly.
So I guess if I understand it correctly, there's no possibility of sharing data from Taiwan. Is that correct?
No. It's still possible for some data.
I see.
We still, but we again, of course, we need IRB to prove that.
Okay.
So, Sardar, one thing we would like to do is have people propose use cases. Then we can take those use cases to the teams that manage a DUA and would be responsible for executing one between the university and Taiwan. So we need some ideas of how we would want to share data with some logistics to propose, just to see if it's feasible. And if the lawyers say no, or if the IRB says no, then we revise our plan and we come back with other ways to share the data and to integrate it.
By use case, you mean projects, studies?
Yeah. How would we do a study?
Yeah, that makes sense.
So I think Yeah, I think, Kayvon said lots of good ideas for collaboration. I think that means we reach out to Kayvon and say, okay, what were your ideas? And Sardar, you and I are going to meet with Nagar, Brittany. We're going to talk about what are our ideas for collaboration. And then we just need 5 or 6 different ideas. And then enough so that we can describe it in a way that a lawyer and an IRB monitor can hammer out a DUA, right?
Yeah, I mean, we talked about the EHR study, but that wasn't. I don't think that we were planning to share data. It was most of like collaborating on the development of the models. But there are, we have some projects that are in the works, are under review, grants, and I don't know what are the implications of using an international site for those studies, because they're under review by NIH. So I've never done collaboration with international sites on an NIH grant, so that's something to find out. But one area that I can see doing collaborations is just to you can use Michigan Medicine as a site for your studies, and vice versa, assuming that there's funding. So that's usually the sticking point, right? So to pay for the people who are actually doing the data pools, running the models, etc. I mean, it depends on the study, but that's probably an area where natural collaboration can happen. Since a lot of the stuff that you went through are pretty mature, they're not new projects. Probably co-development of models probably makes more sense for projects that are at an earlier stage. So yeah, some thoughts. Unfortunately, I won't be able to attend the next meeting. I'm assuming that you were going to present the same slides. Is that correct?
Yes, pretty much the same.
Yeah. The only feedback I can give is if you could condense the slides a little bit so that there's at least 15 minutes left at the end, because that's why you're giving the presentation, right? That conversation and discussion and seeing what people are interested in, finding potential collaborations.
We might have to
maybe
trim a few or separate it into 2 separate presentations. Pick 6 and then another 6 for the second one. That way you can get more into the meat of things, to tell the story about how you began the research, what you discovered along the way, what kind of novel discoveries you have, what you published on. I don't know, Brittany, you're the, among the people in our target audience, at least more along the senior level. What were your thoughts, and how would you approach it to a group of your peers?
I agree with Sardar. I thought all the content was really good. I really liked the end. I really like the specific questions that you focus on the end, and I would probably spend a little bit more time on, kind of your next steps and questions, just to give the audience a little bit more time to digest it, just so we can kind of think more about potential opportunities. But yeah, do
you think a moderated discussion would be the way to go, and have someone who pauses and asks questions in between each topic?
Maybe.
I would say definitely, asynchronous questioning, that would be probably a more fruitful approach.
Yeah.
And also, if you want to focus on things that you think are good collaborations, because things that projects, past projects that are done and over, probably don't. I mean, they may lead to new ideas and new projects, but what you're thinking of right now, what you're working on right now, or things that you're, the digital twin thing. I personally, I'm skeptical, but like that's where it gets interesting. Like it's a future thing that someone may jump on and say, yeah, I want to do that too. Let's do it together. And prioritizing those over things that are maybe in the past, and there's not much to do at this point about.
Mm-hmm.
The causal inference stuff were interesting. The reinforcement, like the last 15 minutes, I think, is where things got really interesting, and like where it was intriguing to think about new projects. So I would maybe make that 15 minutes more of the projects, and maybe remove some of the stuff from the earlier slides.
Mm-hmm.
I do have questions and ideas and things, but like that, we can probably discuss them separately.
Okay.
Yeah, so we can, and since it's just us right now, we can schedule another time. Start our maybe next week is better for you, or
sure.
Yeah, whenever is good before you leave town, to kind of review and talk more, and also just discuss our own opportunities. Since you've got a high level view of everything, maybe you can relay some of this knowledge to Nagar when you have a chance, and the two of you can discuss what you think would be great to have or great to collaborate on. Just shoot the moon on it. If you could have anything possible, what would you want? And then we can use that to kind of inform a proposal for DUA. And then once you and Nagar have had a chance to chat for a few minutes, then we can all get everyone back together again.
Yeah, well, actually, we have the HIV meeting tomorrow, so.
Yeah, I have to move it.
I don't know if
it's not in.
Who's not in?
Nagar's not in tomorrow. I have to. And I was asking you when on Wednesday you might be available.
Yeah, let's move it to next week. I'm pretty busy this week.
All right.
And we can have Kuan-Fu attend as well, since that was one thing that we identified as mutual interest.
Yeah. Okay. Monday's pretty open. We could do like Monday at 10 a.m.
Next Monday?
Yeah, are you in?
Yeah, yeah. Oh,
okay.
Monday
I'm going to add...
unusually empty.
Yeah, I know. I was really confused why I don't have 7 or 8 meetings. So, Dr. Chen, I'm going to add you to this Monday morning invite at 10 a.m. next Monday, and we'll talk about it then.
Awesome.
All right. Well, thanks for presenting. Thanks for getting it together. I think we'll have to just, like Sardar said, we'll have to trim a few of the projects out and focus on the ones that I think are, maybe you could do 1 or 2 case studies on a successful project, but let's focus on the ones that are more emerging projects that the research is forthcoming, it's promising, and we need collaborations on it.
Mm-hmm.
And I think that might be a good way to focus on it.
Okay.
All right. Appreciate it. And
thank you.
I'll try to collect some additional feedback from Mike Schoding and Kayvon and pass it along to you.
Thanks a lot.
Sounds good.
All right. Thanks, everyone. Take care.
Thank you.
Bye.
Thanks. Bye.
Bye.